Good afternoon, everyone. My name is Nick, and I'm an account manager for Azenta in our genomic service division. And it's my great honor to present today's webinar. We have Dr. Kevin Litchfield, and he'll be speaking about non-classical sources of tumor-specific antigens in checkpoint inhibitor responses. Kevin began his career at the University of Leeds, where he did an undergraduate in mathematics before going to do a master's in bioinformatics at the University of York. He then joined industry with Novartis for a number of years before completing his PhD and then postdoc at the Francis Crick Institute, and then went on to get his own lab at UCL. And Kevin, I hand it over to you.
Excellent, thanks Nick, and thanks everyone for joining the webinar. I'll just bring up my slides. So good afternoon. Yeah, the title of this talk is around non-classical sources of antigen and how they may be particularly important in driving response checkpoint inhibitor therapy, as well as also a new source of ideas for future next generation immunotherapies. And just to give the disclosures for this talk, so this all really starts around data you can see here, and as background for people not familiar, we've had a real revolution in the treatment of solid tumors in the last decade with the introduction of immunotherapy and particularly checkpoint inhibitors. You can see here some of the classical data here where we see up to, this is 3 years, and the 5-year data looks just as strong. We see a subset of patients, around 40% of patients, get this remarkable duration of survival where disease stabilizes, and 3, 5, or even longer patients remain disease-free. And this is great, but of course only a subset of patients see these exceptional responses here. This is melanoma. This is probably the best case scenario. We have about 40% of patients have this durable response. So we're particularly interested to understand what differentiates people that do and don't have these exceptional responses, what drives that response, what do T cells recognize in patients' tumors, to try to maybe develop biomarkers to help direct treatment, but also to think about future and new therapies. What can we learn to improve treatment in the future? So our particular interest is the recognition point, the antigens that are presented by tumor cells that are recognized by T cells, and that can stimulate the anti-tumor immune response. And of course, there's a broad range of epitopes that can be presented by tumor cells that can elicit an immune response. Perhaps the most well described are cancer testis antigens. So they date back several decades. Classic markers such as MUC1, MAGE have all been studied for many years, and here you can see some of the historical clinical trial activity where therapeutic vaccination studies have been done widely against these antigen targets. They've predominantly been negative trials, unfortunately, and it's overall believed that these may be poorly immunogenic targets, or they may actually have some potential immunogenicity but not tractable through therapeutic vaccination. And actually many of these same targets are now back under reinvestigation in the context of T-cell therapies. Another classical, well-described type of tumor antigen are those that derive from oncogenic viruses. Well-known examples include HPV, which is associated with a number of cancer types. Here you can see, for example, head and neck cancer data, where patients that are HPV positive have got better overall survival here compared to HPV negative patients. Then importantly, much higher rates of T-cell infiltration in HPV positive disease, supporting the immunogenicity of these epitopes in these tumors. And probably the classical type of antigen that's had the most intense focus over the last decade or so are those that derive from missense mutations in the cancer genome, so-called neoantigens. So a portion of those missense mutations encode mutated epitopes that can drive an immune response. And this has been shown over many years now as important in checkpoint inhibitor response. Here you can see data across a number of cancer types, where if you split patients into a high or low number of mutations, you split the survival curves. Patients with more mutations respond better to checkpoint inhibitor treatment. And this is a topic we've been very interested in for a number of years, and here's a meta-analysis we published last year, the largest study of checkpoint inhibitor response using multiomics data. And in this work, we profiled over 50 different biomarkers in around a dozen or so different checkpoint inhibitor trials. And we found indeed that the biomarker with strongest effect size in predicting positive response to checkpoint inhibitor treatment was the number of mutations, the tumor mutation burden across those tumors, and particularly the clonal mutation burden, so the number of mutations present in every cancer cell that had the strongest effect size in predicting positive radiological response to treatment. And if we look indeed at a patient level here, you can see all the 1,000 patients plotted, and you can see responding patients indeed had around double the number of clonal mutations, so really strong predictor of improved response to this type of immunotherapy. In the meta-analysis, we also saw other critical markers driving checkpoint inhibitor response, key cytokines, for example, CXCL9 expression, that was highly significant, a stronger effect size even than many other signatures that included a number of markers, T-cell inflammation signatures, for example, that have been developed, but they really had poor performance, and CXCL9 individually had a stronger effect size in predicting positive response to checkpoint inhibitor treatment. What was also interesting in this study is the biomarkers that didn't validate. You can see here, for example, well-known biomarkers, for example, the burden of somatic copy number alterations. So this has been previously described as a key predictor of response to checkpoint inhibitor treatment. Higher levels of aneuploidy associated with immune evasion and poor response, and this was published in 2017. And indeed, you can see in these previous papers that the survival curves are different based on the number of somatic copy number alterations, a higher load of copy number alterations associated with poor response in two different cohorts shown here. And we can see that same pattern in our data here in patients treated from another cohort, the Genie cohort of immunotherapy treated patients. It does split survival curves here. You can see patients with high load of alterations have poor survival, but we also see the same pattern in patients not treated with checkpoint inhibitor treatment, but with other types of therapy. We see the same pattern where a high load of copy number alterations associated with poor survival. So it, as always with these biomarkers, there's a difference between what's prognostic, and here this looks like in this situation SCNA burden may be more prognostic, or it could be prognostic and weakly predictive. But in our data set for 1,000 patients, we didn't see a strong association with checkpoint inhibitor response. Another biomarker that we didn't see a significant response in our pan-cancer cohort here was a measure of HLA divergence. So this was published in 2020 here, showing the evolutionary divergence, so how diverse are our set of class 1 HLA genotypes that we inherit, with the idea being a more diverse set of HLA types associates with a greater potential to present different epitope types that might improve the anti-tumor immune response. So we were able to recreate this biomarker, and here you can just show that we obtained identical results as how we derived it, how the authors derived it, but we overall didn't see a strong association for this biomarker in many of the cohorts. We only really saw a positive replication in one particular cohort in the study here that was anti-CTLA-4 treated. So this may be an interesting biomarker where it's specific to a particular type of treatment. And we did see this actually in our formal analysis where we broke down our 1,000 patient cohort into different subgroups of tumor type and drug type combinations. And here you can see that data, and here you can see the one biomarker that we found that was specific only to a certain context. Here for melanoma and CTLA-4 treatment was HLA divergence. And so there may be interesting biology there. For example, prior translational work has showed that anti-CTLA-4 treatment is known to prime a broader set of TCR sequences So potentially that might synergize with an ability to present a broad set of epitopes. So that might explain why that biomarker is strongest in that particular context. The rest of the biomarker landscape, we saw very much the same biomarkers that were significant in the pan-cancer analysis, with the exact same ones that were significant in individual cancer types, tumor mutation burden, for example, CXL9 expression. Many of those were consistent in individual cancer types. We did see some differences in effect size. For example, you can see clonal tumor mutation burden had a much higher effect size in lung cancer, for example, and bladder cancer than it did for melanoma, where it was only really either non-significant or marginally significant. So some difference in the effect and the importance of biomarkers across different cancer types. You can see here also the correlation between biomarkers. Many of these biomarkers are heavily correlated and connected. There's redundancy between them. Measures of mutation burden are correlated, as well as some of the immune cell infiltration markers are heavily correlated. There's a relatively weak correlation between these two separate compartments, suggesting that they could be combined to give a complementary utility in predicting patients for response. And then finally, in this a paper, we also were able to do single-cell RNA-Seq, looking at our local patients' samples recruited at UCL, and to take patients' tumor-infiltrating lymphocyte samples and do a tetramer reactivity sort. This basically measures whether the T cells are reactive or not to a clonal neoantigen. So within the same patients, we get some T cell populations that are reactive and recognize a clonal neoantigen, some that don't, and we can then do single cell RNA-Seq and look at the different markers in each of those two populations. And we saw a real outlier result in this analysis here, CXL13, that was strongly upregulated in the T cells that were reactive to the clonal neoantigen, and also highly upregulated in patients that responded to treatment from the 1,000 patient cohort. So CXL13 looks a very important marker at a T cell level, and also validated in bulk tumor tissue as strongly upregulated and relevant to an effective T cell response to immunotherapy. And so from the summary of that work, we're able to build a basic understanding of the factors that influence checkpoint inhibitor response. We know that the burden of mutations is important, particularly clonal neoantigen burden is important in driving response. We know certain cytokines are critical. CXL9 expression, for example, drives T cell recruitment. That's critical in the immunotherapy response, as well as CXL13. That's critical, promoting, amongst other things, tertiary lymphoid structure formation. But what was really interesting about this work was also how little it could explain. Of all those biomarkers, over 50 different biomarkers we assessed in total, we could only explain half of the variance in response. So when we looked at the proportion of variance analysis, which sort of measures, how much we can explain in who will or won't respond to treatment, despite all this data, whole exome data, or RNA-Seq data, in most cohorts can explain around 40% of the factors that dictated differences in response. So it really suggests there's a lot missing, a lot still to understand. And here you can see that same question a different way here, where we just look very simply at these classical types of antigen, or high mutation burden, or oncogenic virus within the tumor. And clearly those classical types of antigen enrich for responders. They're found at twice the rate in patients that respond. But if we look in our responding patients, still half of them don't fit that category. They don't have a classical type of antigen that explains their response. So we believe there may be a particularly important non-classical types of antigen that are common and explain over half of the responses to checkpoint inhibitor treatment. So that motivated us to look more broadly at other types of antigen that may be important in immunotherapy response that hadn't been fully appreciated. We're particularly interested, first of all, in antigens that derive from dinucleotide variants. So this is where you've got two nucleotides mutated in the exact same position. You can see here they're particularly common in melanoma. You classically see CC to TT mutations in melanoma, linked to UV exposure, as well as other cancer types, bladder cancer, head and neck cancer, also a small number in lung cancer. So these are in important types of mutations found in certain cancer types. And we were interested that, are they a reason, or can they create particular types of neoantigen that may be important and not properly appreciated? So we did further analysis, and here you can see in the first heat map here the types of changes in amino acids. So if you go from a reference amino acid here to an alternative, a mutated amino acid, when you have a single nucleotide change, certain combinations are just impossible. You can't go from a leucine, for example, to an alanine with a single change. It's just impossible based on the codon structure. That cannot happen. But when you start changing two nucleotides, you can have much more different types of changes, a much broader spectrum, as you can see here in the second heat map. Many more unique mutation patterns are possible. In total, we saw here over 100 more unique different types of mutations that came from dinucleotide variants, and a greater shift towards more radical mutations. This is where whole side chains are different compared to single nucleotides that are enriched more for conservative amino acid substitutions. We saw a shift towards a bigger change in Grantham distance, a measure of amino acid difference with dinucleotide variants. And then we also saw a greater shift towards hydrophobicity here with dinucleotide changes. Those epitopes are much more hydrophobic. And of course, hydrophobicity is classically linked to immunogenicity. In a viral context, hydrophobic viral epitopes are known to generate stronger T-cell responses. We're able to confirm this also in the context of tumor neoantigens. Here we see T-cell activity data where we screen different neoepitopes for T-cell activity, and we saw the epitopes that were T-cell reactive were enriched for higher rates of hydrophobicity. Another non-classical type of epitope that we think are important are those that derive from frameshift events. So these have been described by many groups, and here you can see the basic hypothesis of why people think they're important. With a single nucleotide change, of course, you just change one amino acid, and it often may be in a position that is not important. It faces away from the T-cell receptor, so it doesn't create an immune response. You've only got one amino acid that's altered, so it's a lower probability of creating an immunogenic epitope. But when you have a shift in the reading frame, you have a whole stretch of mutated epitopes, 4, 5, 6, or even whole complete epitopes that are completely foreign and mutated and unknown to the human proteome. So you've got a much higher chance of altering an important position, an anchor residue or a TCR facing residue. So you've got a greater chance of creating an immunogenic epitope. We showed this in our prior work that the indel frameshift mutations had a much higher rate of neoantigen generation and enriched for mutation-specific neoantigens. This has also been confirmed in the work of others. Here in head and neck cancer, other groups have shown that frameshift events predict better response to anti-PD-1 treatment. Also in mismatch repair deficient tumors, here in preclinical models from Timothy Chan's group, they showed higher rates of immunoediting and immune elimination of frameshift mutations. So I think they're particularly important. And then a related pathway is the nonsense mediated decay pathway. Anyone familiar with frameshift mutations will immediately think of premature termination codons, and that will immediately bring up the nonsense mediated decay pathway. Here you can see an overview of that pathway. It's an mRNA surveillance pathway that identifies mRNA transcripts that have got PTC mutations, and it degrades them at mRNA level, so it prevents any type of protein being generated, and it may prevent an immune response. So a particular type of non-classical antigen that may be important are mutations that escape this pathway, escape nonsense mediated decay degradation. And we see evidence for this in our patient cohorts here, patients with frameshift mutations that escape degradation through the NMD pathway, higher and higher rates of that in patients' tumors here. Towards the end of this graph, patients with 10 or more mutations that escape degradation, frameshift mutations, have got very high response rates to immunotherapy here, 60 to 70%. And again, these same observations have been found by other groups. Here, Franz Supek's lab in Barcelona identified similar discovery in terms of nonsense mediated decay escape in different methods, different patient cohorts, found the same conclusion. Another non-classical type of epitope that's interesting and may be particularly relevant are epitopes that derive from cancer-associated bacteria. And this was demonstrated very elegantly by work from Yardena Samuels' lab last year, where she showed in melanoma that there were bacterial epitopes that actually present inside tumor cells. The bacteria were found to be intracellular within tumor cells, and that resulted then in class 1 presentation of epitopes from the bacterial proteins presented by the human tumor cells on HLA molecules. And so, of course, that's likely to elicit a strong response. These are non-human epitopes, and that can elicit a strong T-cell activity. We've looked in our data and to see whether do we see evidence for bacteria inside tumor cells, and this is single-cell RNA-Seq data from a collaboration we have with the Samsung Medical Center, and looking here at patients' tumors where we've got tumor cells as well as normal epithelial cells as a control. We can look inside the single cell data, so it measures what bacterial reads are inside the human cells, and we see significant enrichment where there's higher rates of microbial read mapping in tumor cells compared to normal epithelial cells in the same patients. And then another type of epitope, non-classical epitope, that we believe may be important are epitopes that derive from the UTRs, the 5' and 3' untranslated regions of genes. And so, of course, by definition, UTRs shouldn't be able to generate epitopes. They shouldn't ever be translated. But of course, as always in cancer, there's exceptions to these rules, and we see several scenarios where there is translation in UTR regions. For example, where we have stop-loss mutations, where the final stop codon in a gene's lost, and then you see translation that extends into the 3' UTR. You can see situations where there's translation and then another stop codon's reached, and some situations where the whole of the UTR is translated, and then you see a huge amount of mutated epitope sequence that shouldn't normally be present. We see that these mutations can indeed make epitopes. Here they make amino acids that can go up to 50 or 100 amino acids in length, so they can make dozens of potential mutated epitopes. These are also known to be driver mutations. For example, SMAD4 mutations occur through stop-loss mutations, so they can be clonal and frequent across many patients' tumors as they actually drive tumor progression. We also see at the other end of the transcript at the 5' UTR, again, unusual situations where there's mutations that cause unexpected translation. Particularly here, we see upstream open reading frames are created. So this is where we have a mutation, and then it will create a new start codon that isn't normally there, and then we'll then see translation in that 5' UTR. And again, it can either be start and then it stops, and then the normal transcript is translated, or you have out of frame translation that carries from the UTR through to the transcript, or then you can have in frame, but it elongates the transcript. All of those situations will lead to mutated epitopes that potentially are cancer specific and immunogenic and may drive an immune response. So we've begun to look at this in our lab in unpublished data shown here, where you can see each of those situations, the normal frameshift mutations that come from coding mutations, and then the start gain mutations at the 5' UTR, and the stop-loss mutations at the 3' UTR. And you can see here they all create mutated epitope sequence here. Particularly the start gain mutations create a higher number, around 25 to 30 in terms of the length of mutated epitopes here, shown across two different whole genome sequencing cohorts. And when we look at whether this predicts response, does it associate with better response, checkpoint inhibitor treatment, we can see here that whereas these mutations get longer, as the mutations create more and more mutated epitope sequence, we see a stronger effect, where the particularly long epitopes here, where start gains that generate over 50 amino acids of mutated sequence, have a particularly strong association with improved rates of checkpoint inhibitor response. So just to, in the last part of the presentation, I just want to share some of our functional work. So analysis of patient sequencing cohorts is of course very powerful because it's, ground truth directly in patient samples, but it's of course only correlative in nature. So we also wanted to try and take some of these observations back to the lab and then do functional experimentation to see which types of non-classical antigen have got experimental evidence confirming their importance. So collaborating with another lab here at UCL through James Redding's lab, also with Sergio Quesada's lab, we're able to do the manifest assay, which is shown here. This is an assay where you just simply do T-cell receptor sequencing of a pool of T cells before and after you stimulate those T cells with a pool of epitopes. And then when T-cell receptor sequences expand rapidly after stimulation, it suggests reactivity or recognition of epitopes in that pool. So it's a good way to measure which epitopes actually are T-cell reactive and drive expansion of T cells. And so in our preliminary work here in this a cohort, we can see here the first patient we've done. Here we see reactivity to a number of these non-classical types of epitope, some of the viral microbial epitopes, as well as some of the frame shift NMD escape mutations. And just to show this data in depth, the real benefit of this approach we've done here is the ability to collaborate with the rapid autopsy study we have here at UCL, the PEACE autopsy study that's led by Mariam Jamal Hanjani and Charles Swanton, and this patient actually kindly consented to have autopsy tissue sampled, and so we're able to look at the T-cell receptor sequences present across all their different sites of disease. This patient had a primary lung tumor, but then had lymph node metastases, liver mets, bone mets, brain mets, and so we could look globally at the T-cell expansion across all those sites at the time of autopsy. And so here you can see two TCR sequences that really stood out as globally expanded, present in all the sites of the primary tumor, as well as all the sites of metastatic disease. And when we link those back to the functional manifest T-cell activity screens, we see those TCR sequences recognize two different types of epitopes. The first one here, a dinucleotide, a variant, and the second one there, an epitope that comes from a frame-shifted NMD escape indel. So it's only, I guess, an N of 1 patient, but in this patient, at least, we see the non-classical types of antigen as strong drivers of T-cell response globally across all their sites of disease. So just to give some summaries from this work, we see a real complex set of biomarkers that associate with checkpoint inhibitor response. In our recent pan-cancer meta-analysis, we see mutation burden, particularly clonal TMB, as the strongest driver of response to checkpoint inhibitor treatment, but also CXCL9 expression is critical, driving T-cell recruitment. CXCL13 expression is important, associated with tertiary lymphoid structure formation. However, we find even across all those data sets, only half of the variants can be explained. So there's a number of key factors still undiscovered that determine checkpoint inhibitor response. We think one of those factors may relate to non-classical types of epitope, in particular epitopes that come from dinucleotide variants, as well as frame-shifted variants, and as well as bacterial epitopes described in the work of others. In our preliminary functional validation experiments using the manifest reactivity assay, we see support for non-classical epitope types as having potential to drive strong T-cell response. And really we're motivated to try to explore this further to come up with new therapeutic approaches to exploit these non-classical types of epitope. They may be particularly important in low tumor mutation burden cancers, where they don't naturally have a high number of mutations. And this covers many types of cancers, common cancers, colorectal cancers, breast tumors, prostate tumors. They're all commonly low tumor mutation burden. It's particularly important to develop new therapeutic approaches to enhance and exploit non-classical types of epitope. And we're particularly interested in this context in childhood solid tumors. So we've recently been awarded a Grand Challenge, Cancer Grand Challenge funding through a huge team across UCL, also Washington and Paris, an international team working led by Martin Poulet and Catherine Ballard to develop new CAR T-cell therapies for childhood solid tumors. And of course, those are all low mutation burden, those tumors. So non-classical types of epitopes will be important in developing new treatments for this urgently needed clinical context of childhood solid tumors. And we've got funding if anyone's interested in terms of job opportunities. We've got 5-year postdoc funding for a bioinformatician to lead the analysis for this whole project. So it'll be really exciting data and a really worthwhile clinical context. So please email me if there's any candidates interested in that opportunity. So just to finish with the acknowledgements for this work, to thank my lab at UCL, the TIGI lab. So thanks everyone for all their contributions. Also to thank our collaborators, particularly Sergio Quesada here at UCL, as well as Charlie Swanton at the Crick Institute, and to thank our funders, the Cancer Research UK Lung Centre of Excellence, as well as the City of London CR UK Centre, as well as my funding from the Medical Research Council. And thank you for your attention, and happy to answer any questions.
Great, Kevin, thank you for that. That was a really interesting talk. We actually already have a question in the chat, and just a reminder, if you have any further questions, please do ask them in the chat, and then I can ask them to Kevin. So the first question from the chat says, when looking at biomarkers with your 1,000 patient cohort compared to previous studies that did align, are the makeup of the cancer types relatively similar, or could some of the differences be explained by different makeups of cancers in those studies?
Yeah, that's an excellent question. We had bladder cancer, melanoma, lung cancer, as well as a pan-cancer solid tumor cohort of around 10 other different cancer types. So we did have fairly broad coverage of the cancer types. And so all we can say really from our analysis definitively is what biomarkers are important at a pan-cancer level. So there could be additional biology as cohorts grow in size, as we get thousands of patients in each individual cancer type. That's what we really want, to be able to definitively say, you know, what's important at a tumor type level. Where we were well powered in things like melanoma, bladder cancer, we had over 300 tumors or more. We didn't see huge differences, to be honest. There wasn't great evidence of tumor type specific biology, but we can't conclusively say, yeah, but it doesn't look strongly tumor type specific, but we need future work to be confident on that.
Great. And I actually had a question for you. So when you're looking at your kind of mutations in the 5' UTRs, have you done any ribosome footprinting to kind of see if you're actually seeing translation at these new start codons, or if they're just being transcribed?
No, that's a great question. Yeah, we haven't had that data. Yeah, we've been really working hard to get whole genome sequencing data first, whether checkpoint inhibitor response. In terms of ribose-seq, we haven't had any data generated in those sort of cohorts. We haven't done it yet, but we are working to try and combine ribose-seq data from other, you know, sort of reference databases. And I know there's actually, I think, even today published a new atlas or experimental data set for ribose-seq cohorts. So that would be helpful to look and to validate whether there's actually translation at these regions. I mean, in terms of upstream open reading frames, that they are common and well described. as a normal biological process to regulate protein expression. So they're functionally validated, so we would expect that they are, translated, but we are trying to further validate in a cancer situation where there's new mutations that make new upstream open reading frames. Are they translated with riboseq data, but then also ultimately do we see protein evidence of them? So we've got proteomic data sets, we're trying to find mass spec proof of those peptides, as well as then HLA peptidomics to show HLA presented epitope proof of those events.
Great, and that leads me nicely into another question we have. So you've already demonstrated the power of kind of multiomics and kind of laying all of these data sets over top of each other to continue to learn more and more. What kind of additional layers of kind of omic-based data do you think would kind of ideally you'd like to see kind of next?
Yeah, I think there's probably two big gaps. The spatial aspect, so everything at the moment is unspatial, so we're blinded to patterns of co-location of tumor cells with different immune cells together, and fibroblast populations. So emerging technologies, such as whole transcriptome sequencing on spatial transcriptomics platforms, those will be, I think, particularly insightful to look at spatial patterns, as well as, I think, our work so far, a lot of work in the field has been tumor-centric. So it's been zooming in on the tumor tissue, but really missing host-based biomarkers. So looking at PBMCs, profiling TCR patterns in peripheral blood, that might be particularly insightful, as well as measurement of cytokine levels in peripheral blood. There, I think, would be an important gap that would add to this question.
Great, and I have one final question at the moment that's come in, and this is about splice factors, and saying that some of the non-classical sources seem to be related to splicing and splice factors. And do you think that this is kind of potentially a driver for cancers in some of these non-classical sources?
Yeah, I think it definitely would be related to splicing errors, and there was a paper published by Memorial Sloan Kettering. They published a paper actually a year or two ago where they showed inhibition of a splicing factors enhanced epitope presentation, and maybe a way of enhancing anti-tumor immune response. So definitely splicing defects where they occur naturally, or if therapeutically, if you can start to impair splicing, would be part of the upregulation of non-classical epitope presentation.
Great. Well, thank you very much, Kevin, for taking the time to present to us your wonderful work, as well as answering the questions. I currently don't have any further questions, so I'd like to thank everyone for. Oh, apparently we have had some further questions come in. We have. My apologies. Given that, so another question that came in was, given that TCR generation is highly regulated with positive and negative selection, ensuring that only things that can be transcribed generate cognate TCRs, at least that is according to the textbook. Does your data of TCR against UTRs, for example, mean that there's more to the story about how TCR generation happens?
Gosh, that's a great question. I guess what we've looked at in terms of TCR sequencing in our manifest data, we've not, I guess, sort of looked at them more broadly in terms of TCR selection and central tolerance. As data sets grow, and we're trying to work in collaboration with others, generate larger databases of cancer-associated TCRs from tumor tissue and peripheral blood of cancer patients. As those data sets grow, that will be really to look back and see if that can sort of understand or inform about TCR selection. So we haven't understood that yet, but that's a good question.
And another question that has come in, if I can scroll to find it again. with some of your sequencing findings, what percentage of your 1,000 samples show a specific mutation to call out as being of interest?
Yeah, that's a good question. I mean, I guess it depends what you classify as a mutation of interest. I mean, things like, hotspot recurrent mutations in things like KRAS G12 mutations, or recurrent P53, missense mutations. I mean, they are common, and when you look collectively across sort of a cocktail of mutations like that, you can get fairly high number of patients. 30-40% or more patients may have recurrent mutations in those sort of classic genes. And I guess one of the translational applications of this work is around non-personalized off-the-shelf approaches. The great challenge of most mutations is they're private, found only in each individual, and that makes it very difficult for cost-effective generation of immunotherapies. So of course we see 99% or more of the mutations are private one-offs, but an interesting number, getting towards half or more patients might have recurrent hotspot mutations. And then in terms of the non-classical types of mutations, the stop loss mutations or the start gain mutations, some situations like SMAD4 and other situations they are recurrent because they're drivers, but in many other situations they're private mutations. So that is one challenge with this work, is how do you translate it into a cost-effective therapy? So that's an ongoing thing we're exploring.
Yes, and that leads nicely into, I guess, kind of another question, which is kind of a very broad question, but how will your research impact possible changes in cancer treatments?
Yes, so I guess there's two areas. The biomarker context. So as you saw at the start, only around 40% of patients see this exceptional benefit, and so identifying that subgroup at the start would help, because then the 60% can go into trials or novel types of treatments. If they're not going to drive benefit, then it's good to know that up front. So if we can better stratify patients, and we've got start of a prospective trial to validate a biomarker set of a number of the things from that paper, a multivariable biomarker that we hope may be accurate in terms of stratifying patients that will and won't respond, and then that may benefit patients that won't respond to go on to a different treatment that does benefit them. The second type of area is obviously novel therapeutics, next generation of immunotherapies, either personalized vaccine or T-cell therapy approaches, or new ways, small molecule ways to try to alter these processes that we think may be important in epitope generation.
Great, thank you. So we now have actually answered all of the questions that have come in. And so I would like just to thank you, Kevin, for giving your time again, for answering questions and for presenting your work. And I'd like to thank all the audience for joining in. There's going to be a follow-up email with a link to this webinar for anyone who'd like to view, or for anyone who didn't get to join right at the beginning, if they'd like to re-watch that. And yes, thanks again.
Excellent. Thanks, Nick. Thanks, everyone. Bye-bye.
Bye.